Hi Year 12s. In this video we're going to go through together the structure of DNA. Now the reason we're doing this is because in the study design, VCAR have asked you to understand what nucleic acids are for information molecules that encode for instructions for the synthesis of proteins. So that's kind of the bigger picture, but today's video is mainly focused on understanding the structure of DNA. We will then take that forward and use our understanding of DNA to inform our understanding of RNA, and then how that fits that bigger picture in terms of protein synthesis. So you'll know that you're successful in this lesson if you can define the term nucleic acid, if you can draw and label the structure of a nucleotide, if you can explain the structure of the DNA double helix, including the hydrogen bonds that hold those bases together, if you can apply those base pairing rules, and if you can model the impact of the anti-parallel nature of the double helix. That last dot point, we'll unpack that further in the classroom. So let's start with what nucleic acids are. Nucleic acids are biomolecules, and they're essential for life. So they consist of both DNA and RNA. They're a polymer, so poly means many, which means they're made up of many repeating units. So they're a polymer of what we call monomers. So mono means one. I always remember that because someone with a monobrow has one eyebrow. So a monomer is a singular unit, and a polymer is when we have many of those joined together. And they essentially contain the instructions for the cell, instructions for proteins essentially that are essential for our cells' regular functioning. So we will unpack RNA in the future, but this side here, this DNA side, is what today's lesson is focused towards. So I guess in the bigger picture, this molecule here that's kind of being unwound, this is a DNA molecule. It's contained in the nucleus in the form of a chromosome. This is for a eukaryotic cell, I should mention. That DNA, when we're looking at protein synthesis, if you remember last lesson, you looked at the structure of a cell. DNA is in the nucleus, but the thing that I guess converts that message into a protein is the ribosome, and that's all the way out in the cytoplasm. So mRNA or RNA is part of that messenger molecule that takes that instruction out of the nucleus into the cytoplasm to be transcribed and translated into a protein. Now let's hone in just on DNA for now. So DNA stands for deoxyribonucleic acid. It's a double helix molecule, and when we say double helix, it's this fun twisted shape, and we'll talk a little bit later on why it's so twisted. But basically, it's the unit of inheritance for all living things, and one segment of that DNA is what gives us a gene, and a gene is a segment of DNA that encodes for a protein or some sort of gene product. So let's hone in on this picture here to start with. You can see here that it's made of this regular repeating unit. That regular repeating unit, that is our nucleotide. Each of those is identical except for this part here. That's our nitrogenous base. Now I say it's a nitrogenous base because it's a base that's got nitrogen in it. Once we pack them together, we end up with this ladder looking molecule that in real life is quite twisted into this double helix shape. Okay, so let's start with the nucleotide now. So your textbook and lots of pictures show a nucleotide drawn like this, a circle that represents the phosphate group. You can see that over here. A sugar molecule, and then normally a rectangle or some sort of funky shape that I guess demonstrates this nitrogenous base. And here you can see all the little nitrogens that are embedded within that base structure. Everything stays the same always, except for that nitrogenous base, depending on which base we are looking at. It could be adenine, guanine, thymine, or cytosine. And we'll unpack that further in the next few slides. The reason I've shown you this I guess, chemical structure of the nucleotide is that it's important to understand that this sugar is made of carbon atoms. So each corner here is a carbon atom. So we number it starting here from the first one that's connected to the nitrogenous base here. And we just go up from 1, 2, 3, 4, and 5. Even though this carbon's not included in that ring, it's still a really important carbon in the nucleotide structure because it helps with the direction of our backbone of that ladder. So let's unpack the nitrogenous bases a little bit further. So you can see there's nitrogens all over this page here, and those nitrogens those nitrogens make up the bases, and we can classify them as two different types. They can be either purines or pyrimidines. Purines are basically a base that's made up of 2 nitrogen rings, and you can see each of those has 2 rings involved, whereas the pyrimidines just have one of those. Most of you will remember from previous years the base pairing rules. That's thymine, and let's ignore uracil for now because that's part of RNA. Thymine always bonds with adenine, and cytosine always bonds with guanine. So basically one of these double-ringed pyrimidines always bonds with a single-ring purine. The other thing that I want you to point out here is that these bonds in between these nitrogenous bases, they are called hydrogen bonds. They're a really strong bond that holds DNA really tightly together, but it's weak enough for it to be able to be ripped apart for the message to be translated and then joined back together. There are 3 hydrogen bonds between guanine and cytosine, and there are 2 hydrogen bonds between thymine and adenine. So now that we've unpacked the nucleotide as a monomer, let's look at the DNA structure as a polymer. So if we have a look down the left-hand side, you'll see there's that repeated pattern of sugars and phosphate, sugar, then phosphate. That's what we call our sugar phosphate backbone, because it's essentially the backbone of the molecule made up of repeating sugars and phosphates. Between that sugar and that phosphate, as each nucleotide, I guess, is combined to the next one, we have what we call phosphodiester bonding. Between each nucleus, if we're now looking at 2 different strands, within them you can see those hydrogen bonds. So between adenine and thymine, we've got 2 bonds. Between guanine and cytosine, we've got 3 hydrogen bonds. Now, the last thing that we need to have a look at is this anti-parallel 5' to 3' nature of the DNA molecule. And you can see that here, if we're moving up on the left-hand side, we're going from 3 to the 5' end. And on the other side, it's going in the opposite direction. And what that means, if we go back to those numbering of the carbon molecules on that sugar, that if we start here, if we're looking at this adenine molecule, this is our first carbon, second, third, fourth, fifth. And the bonds are occurring on the 3rd and the 5th carbon, moving upwards. Same here on the one underneath, 1, 2, 3, 4, 5. So all of those carbon molecules are oriented in the same direction. On the other side, they're upside down. So if we start here on the thymine, 1, 2, 3, 4, 5, you can see quite clearly that that's shifted upside down. And what that does is it creates almost a crooked ladder. So we draw the ladder really nice and normal and straight when we're drawing images of it. But in real life, it's a little bit slanted, and that causes that twist of the DNA molecule. And you'll have a chance to model this in class. But essentially every 10.5 base pairs, the DNA molecule goes through a full twist. So it's a full 360 turn because of that 3 to 5' anti-parallel nature. Making it a little bit crooked, causing it to twist around. Now let's practice an exam style question. I want you to read through and give that your best answer. Hopefully you're able to identify that the macromolecule in group B is a DNA molecule and that a monomer of that is made up of a deoxyribose sugar, a phosphate group, and a nitrogen containing base. Let's have a go at this one. So have a read through and select what you think the answer might be. So the answer there is C, and that's, I guess, applying and practicing your base pairing rules. So C always bonds with G, G always bonds with C, A, T, and so on and so on. And that gives us this answer C. Last question, well, it's a double whammy, but this is from VCAR 2015. So you can see there that a single nucleotide is this whole unit here. So that contains all X, which is the nitrogenous base, Y, which is our sugar, and Z, which is our phosphate. The second question, the feature of DNA that can be seen in this diagram is the anti-parallel arrangement of the two strands of nucleotides. And we can tell it's anti-parallel because it's been labelled with the 5' and 3' ends.